Hi, good afternoon, everyone. So my name is Aaron Hoskins, and I'm a professor here at University of Wisconsin-Madison, representing the RNA MaxiGroup. And it's my pleasure to introduce to you three of our great graduate student trainees here at UW-Madison, who will be telling you three stories about their research. And so our first speaker is Karli Lipinski. She is a graduate student in the Department of Chemistry and a member of my own laboratory, and she'll be telling you about her work on splice site selection. And so with that, Karli, take it away. Oh, I just want to remind everyone too that if you have any questions, you need to ask them using the Q&A box, and then I will moderate the questions at the end of each talk. Thank you.
Thank you, Erin, for the introduction, and thank you to the organizers for inviting me to speak today. I'm very excited to present my work on the splicing factor F5V6 and what we know about how it regulates 3' splice site selection. So splicing is an essential process for eukaryotic gene expression, where introns are removed from pre-mRNA transcripts. The spliceosome catalyzes this reaction and is a large protein and RNA complex that also recognizes the boundaries of the intron. Here, the 5' splice site and branch site are recognized by components of the spliceosome called the U1 and U2 SNRPs. And the first step of chemistry produces a free 5' exon as well as a lariat intermediate. And the second step of chemistry produces the mRNA product, both of the exons having been ligated together, as well as the lariat. And so if you recognize the consensus sequences for the 5' splice site, the branch point, and the 3' splice site, you'll notice that the consensus for the 3' splice site is much fewer nucleotides than either of the other sequences. And so it's been a question in the field for a long time how the 3' splice site is being recognized during splicing by the spliceosome. And so what we know is that second step factors that join after the first step of chemistry has been completed help with stable docking of the 3' splice site within the spliceosome active site. And those factors include SLU7 and PRP18, which work together with PRP8 for that stabilization. The second step factor, PRP22, then translocates on the 3' exon to facilitate proofreading of the stability of the docking within the active site. And so joining the ranks of these second step factors is a newly discovered splicing factor called F5V6. And so prior to our work on F5V6, which was published in 2023, very little was known about the function of F5V6 at all. F5V6 is a non-essential protein in yeast that's relatively small, and it was known to be nuclearly localized. When we delete F5V6 from the yeast genome and spot the yeast at various temperatures, we find that at temperatures other than 30 degrees, yeast lacking F5V6 grow extremely poorly. We did a number of experiments to determine that F5V6 was acting at the second step of splicing. And when we were looking at how deletion of F5V6 is affecting splicing of endogenous genes, we happened upon an interesting result in the splicing of the two intron-containing genes Sus1. So here I'm showing RT-PCR where we can see on this gel bands for the pre-mRNA of Sus1, the partially spliced product where the second intron has been removed, and the mRNA product. However, we find that when we delete F5V6, there's production of an alternatively spliced mRNA product. And when we isolated this product and sequenced it, we found that an alternative 3' splice site was being used within the intron much closer to the branch point. And so this work motivated us to look globally across the yeast transcriptome to see how introns or splicing was being affected. And so we performed an RNA-seq experiment in yeast background where the nonsense-mediated decay factor UPF1 had been deleted. and FYV6 had also been deleted in one of the strains we sequenced. We selected for RNAs that had been polyadenylated and searched for non-consensus splice junctions by aligning our RNA datasets with the splicing aware aligner HiSat2. We then, for example, were able to identify alternative 3' splice sites in these alignments, in addition to our canonical site. And to determine how these sites were being used in our FYV6 deletion strain compared to a wild type strain, we counted the number of junctions or splice junctions, number of reads within our alignment corresponding to splice junctions, all using the same 5' splice site, but using either the canonical or one of the two alternative 3' splice sites, or one of the many in our data set. We're then able to calculate a measure called fraction of annotated splicing, which is a ratio of the alternative junction counts for a specific alternative junction divided by the number of canonical junction counts. And we would expect that if we had a larger FANS value, we would have more alternative splice site usage for that particular junction. Now we can plot our calculated FANS values where the FYV6 is present on the x-axis and absent on the y-axis. And when the FANS values are equal between presence and absence of FYV6, we would expect the points for FANS values to cluster along the dotted line. However, if usage is higher in the FYV6 deletion, we would expect that the FANS values would be on the top portion of the dotted line. And if the junctions were less frequently used in the deletion, they'd be on the lower portion of the line. We additionally colored the points by whether or not the 3' splice sites were proximal or internal to the intron compared to the consensus, or distal internal to the exon. And based on what we know about sus1, we would expect that there would be more proximal 3' splice site usage in these alternative sites in our FYV6 deletion. And when we look at our data, that's exactly what we see. We see more pink dots above the dotted line, so they have higher FANS values in the FYV6 deletion. And so one of the factors that we wanted to look at based on this data is how close are these proximal 3' splice sites to the branch point. And so we plotted a ratio of FANS for FYV6 divided by FANS for wild type. It's flipped on this slide. And you can expect in our graph that higher usage of these alternative sites in our FYV6 deletion would be higher up on the graph, and lower usage would be on the bottom of the graph. And additionally, on the x-axis, we have calculated the distance from the canonical branch site for each of our alternative splice sites that are activated in our FYV6 deletion strain. And so since the sus1 has a splice site that's much closer to the branch point, we were kind of expecting to see a cluster of data here where the distances are much closer to the branch point. And so that's exactly what we see, is we see a cluster of alternative splice sites that are more preferentially used in our FYV6 deletion from about 7 to 20 nucleotides. And this motivated in vitro work with a reporter gene where we varied the distance from the branch point to the 3' splice site. And when we do a primary extension assay and look at mRNA, the amount of mRNA that is produced, as well as the intermediate product, the 3' lariat, we can see for shorter branch point to 3' splice site distances, it doesn't make a difference whether FYV6 is present or absent. These are all spliced quite efficiently in the second step. However, when we increase the distance between the branch point and 3' splice site, we see a marked decrease in the second step splicing efficiency when FYV6 is absent. And so we're thinking that FYV6 is helping to facilitate efficient splicing of introns with longer branch point to 3' splice site distances. And we were really interested in mechanistically how this is being accomplished by FYV6. And again, not much was known about the protein, and so we had very little information to go off of. And so we were greatly aided that after we had published our work, our first paper on FYV6, we were contacted by Max Wilkinson, who had a very nice P-complex structure or product complex of the yeast spliceosome at a very high resolution, or 2.3 angstroms. And you can see here in the structure, FYV6 is this pink elongated protein composed of 3 alpha helices. And what was initially very interesting and striking to us about this structure was the extensive interaction that FYV6 has with PRP22. And so here's a close-up of the structure and the N-terminus of FYV6 interacting with PRP22. Boxed in is a small hook region, as well as the first alpha helix of FYV6 that are making these extensive contacts. This hook region of FYV6 was very interesting, given that the sequence conservation is present across yeast to humans, with the human homolog of FYV6 being FAM192A. And so based on the structural data, we were very intrigued by this, and we were wondering if this interaction with PRP22 was important for FYV6 function, since PRP22 helps to proofread the 3' splice site. And so to examine this a little more closely, we truncated various regions we thought might be functionally important in FYV6, starting with regions that contact PRP22 at the N-terminus, right before the hook, through the hook, and then through the first alpha helix. We also made several C-terminal truncations where we truncated through a portion of FYV6 that's not resolved in our P-complex structure, and additionally through a region that interacts with the 19-complex protein CYP1. We can then express these truncations in yeast and see if they're able to rescue the growth phenotype observed upon the deletion, or if these truncations resemble the deletion, meaning that they would potentially be functionally important regions for FYV6. And so we look at our N-terminal truncations. We can see that when we truncate the regions that interact with PRP22, we only get very modest growth defects, especially in the second one, where we have truncations through the first alpha helix of FYV6. However, when we truncate through the CYP1 interacting region, we get, again, these marked growth defects that resemble the FYV6 deletion in its entirety. We can then look at splicing specifically by seeing if the alternative mRNA in Sus1 is produced in each of these truncation strains. And we find that for the truncations through the CYP1 interacting region, as well as portion of the C-terminus of FYV6, we do see an appearance of this alternatively spliced mRNA product in Sus1. And so our growth data correlates very well with our splicing data for production of this alternative mRNA. And so we think that this interaction with CEF1 is functionally important for FYV6. We can therefore do the complementary experiment where we delete or truncate the FYV6 interaction region in CYP1 to see if we can recapitulate the growth phenotype for the FYV6 deletion. And what we see when we express these CYP1 truncations in a strain where full-length FYV6 is present, we can see that when we truncate through the FYV6 interacting region, we begin to see growth defects that resemble the FYV6 deletion in its entirety, which are then exacerbated when we further truncate through the C-terminus of CYP1. Again, looking at our Sus1 RT-PCR, we can see that the appearance of this alternatively spliced mRNA occurs when we truncate through the FYV6 interacting region in CYP1. And so we think that this region is functionally important for FYV6, or alternatively important for recruitment of FYV6 to the spliceosome. Now I'd like to take a moment to combine what we know about FYV6 3' splice site selection with our cryo-EM data. And so FYV6 is this elongated protein forming these multiple contacts across the spliceosome. It does bridge over the active site and a portion of the intron, including the region from the branch point to the 3' splice site. Since introns with longer branch point to 3' splice site distances are more affected in the deletion, a potential possibility that could be in the absence of FYV6, these regions are less constrained and more easily able to move about, which may disrupt the stability of the docking of the 3' splice site within the active site. And so, yes. And so just to quickly summarize what we know about FYV6 and how it's affecting 3' splice site selection, we know that when we delete FYV6, we get a dramatic cold and temperature growth phenotype, as well as changes to the endogenous gene sus1 or splicing of the gene. When we look globally, we see activation of proximal non-consensus 3' splice sites in the absence of FYV6. We can see that FYV6 is present in spliceosomes as we have a very high resolution structure of the yeast P-complex structure or P-complex spliceosome that contains FYV6. And finally, one of the functionally important regions we've been able to identify in FYV6 is this interaction with CYP1. And so we still have a lot of questions that remain about FYV6. The first being the nature of the interaction between FYV6 and PRP22 and whether or not FYV6 has any influence on PRP22 proofreading activity. We also want to know the nature of the FYV6-CYP1 interaction and how that's influencing the function of or recruitment of FYV6 to the spliceosome. Finally, since FYV6 does have this marked growth defect at higher and lower temperatures than 30 degrees, we're wondering how splicing is affected globally in these FYV6 deletion strains. And what I'd like to leave you with is that FYV6 is this non-essential and for many years very uninteresting protein that turns out to have a very kind of interesting effect on splicing. And how many other non-essential proteins of unknown function are there that could potentially be splicing factors that we might be missing? And so I would like to thank Aaron Hoskins for his mentorship during my graduate work, as well as Dr. Kathy Sen, who was integral to making this project a success. And we worked very closely together on this project, as well as a few talented undergrads, Natalie Zepps, Amory Griffin, and Tyler Chang, who worked on various aspects of this project. And a huge thank you to Max Wilkinson, who provided us with the amazing P-complex structure showing FYV6. We are incredibly grateful. I'd like to also thank alumni that have passed through the Hoskins lab, as well as our funding and resources we've used for the RNA-seq work at UW-Madison. Thank you so much, and I'm happy to take questions.
Great. Thank you, Karli. So we have time for like 1 or 2 questions. And so the first question is from Sam Butcher. Did previous cryo-EM structures have unassigned density corresponding to FYV6, or was the resolution too low to observe it? Is there density for FYV6 homologs in other spliceosome complexes?
So yes, so we have looked at unassigned density in several yeast spliceosome structures, particularly in the C-complex structure. There's very compelling evidence that FYV6 exists as a part of that structure. It's kind of a very similar shape and a very similar location. However, that structure was not high enough resolution to see individual amino acids of FYV6, and so therefore the protein could not be localized on that structure. As far as human spliceosomes, so the human homolog of FYV6 is FAM192A, and that's present in the human pre-C star 1 and 2 complexes that were published in 2022.
Great. Thank you, Karli. So I think we'll go ahead and move on to our next speaker.
Thank you.
Great. So our second speaker is Lauren Mazurkiewicz, who's a graduate student in the IPIP graduate program here at UW-Madison and is a member of Amy Weeks's laboratory. So take it away, Lauren.
Okay. Video. All right. Thank you for the introduction, Aaron. As Aaron mentioned, I'm a member of the Weeks Lab, and the Weeks Lab primarily focuses on protein engineering and proteomics, but over the course of my graduate work, I've come across some interesting things regarding translational fidelity. And so I'm excited to share this for an RNA audience for the first time and to hear what insight you all might have. So my research focuses on a unique post-translational modification called diphthamide. And diphthamide is known to only occur on one protein in the cell, elongation factor 2, which is the GTPase responsible for translocation of the ribosome during translation. And diphthamide is a pretty complex post-translational modification. It takes 8 different enzymes, DPH 1 through 8, for its biosynthesis, and it takes a series of different steps to get to this final modification here called diphthamide. And so even though diphthamide is conserved amongst all archaea and all eukaryotes, its function is not very well understood. And here I show you the structure of elongation factor 2 from S. cerevisiae. And diphthamide can be found way at the tip of domain 4 here. And domain 4 is located directly in the A site of the ribosome, where diphthamide makes extensive contacts with both the mRNA transcript and the decoding tRNA. And based on these initial structural studies, it's been hypothesized that diphthamide is important in maintaining translational fidelity, and particularly in preventing -1 frame shifting, where diphthamide located in the A site interacts with both the mRNA and tRNA, and during translocation prevents the mRNA from slipping back. In the absence of diphthamide, the mRNA is more likely to slip backwards, causing a -1 frame shift. And there have been some initial studies that have supported this hypothesis. For example, a paper published in 2021 computationally predicted various mRNA slippery sites throughout the yeast transcriptome. And they then went on to test a few of these sites in yeast that either had diphthamide or had diphthamide knocked out. And they found that lack of diphthamide increased -1 frame shifting for several different proteins. However, this was only tested on a small subset of the proteome, so it was still unclear how much this frame shifting actually occurs. A more recent study published in 2023 conducted ribosomal profiling in wild-type and DPH2 knockout yeast. And what they found is that there is an increase in the ribosomal drop-off rate in DPH2 knockout yeast. And this indicates that the ribosomes are encountering out-of-frame stop codons, which indicates that frame shifting is occurring at a higher rate. And so while this gave broader insight into the translatome, it didn't really say how much the actual protein products or the proteome was changing in response to this increase in frame shifting. And so this brings me to the main questions that I wanted to address in my research. And that was, to what extent does diphthamide influence the cellular proteome? And does diphthamide affect the translation of certain proteins more so than others? And to do this, I was taking a quantitative proteomics approach using mass spectrometry. And to go through this workflow, I grew yeast that either had diphthamide knocked out or were the wild type strain. I labeled protein extracts from each of these strains, and I'll get more into the labels on the next slide. And these protein extracts were then run on the mass spec, and I could then quantify the amount of each protein in each strain and identify proteins that had significant changes in their abundance upon diphthamide deletion. And so the proteomics approach I used is called tandem mass tag or TMT label proteomics. And this essentially allows you to pool together 11 different samples at the same time. And so these labels chemically modify the N-termini and lysines of peptides. And each of the 11 different labels contain different distributions of isotopes along the label. And these isotopes add up to the same mass. However, during a mass spec experiment, fragmentation occurs in the mass spectrometer, and the labels will then be cleaved. And this causes each of the 11 different tags to have different masses, so I can quantify how much of that peptide was in each of my different samples. So in the context of an entire proteomic workflow, this involves growing my yeast, extracting the protein, digesting it with trypsin, and then I can label that particular strain with a particular TNT label, pull together the protein from 11 different samples where each sample has a different tag. And then a protein will all have the same mass during the first round of mass spec for a given peptide. And then after fragmentation occurs, the tags will have different masses, and then I can quantify the amount of that peptide in each of the 11 different samples. And so this is important because there can be quite a bit of variation between different mass spec runs. And so by having all of my 11 samples in the same mass spec run, I can improve my quantification accuracy. And so here were the results where I compared the DPH2 and DPH3 knockout proteomes compared to the wild type yeast proteome. And what I found was that there was not widespread changes in the DPH knockout strains compared to the wild type, but rather select proteins had significant changes in their abundance. And so this supports the hypothesis that diphthamide affects the translation of certain proteins more so than others. And when I was looking through the significantly altered proteins, what immediately jumped out to me was that a lot of them were yeast retrotransposons. And yeast retrotransposons are portions of retroviral genomes that have inserted themselves into the yeast genome over time. And these retrotransposons contain +1 frameshift sites, where when frameshifting does not occur, only the GAG portion of the polyprotein will be translated. But when +1 frameshifting occurs, both the GAG and the poly portions of the polyprotein are translated. And so when I looked more at my results, what you can see here on the y-axis is the protein abundance ratio comparing the DPH2 knockout with the wild type. What you can see is that the non-frameshifted form of the retrotransposons were increased in the DPH2 knockout strain relative to the wild type. And so this was interesting to think about in the context of prior work, which is mostly focused on -1 frameshifting and not +1 frameshifting. And this has allowed me to form kind of a new hypothesis regarding diphthamide and program +1 frameshift sites, where diphthamide in the A site may prevent the mRNA from sliding backwards. It doesn't necessarily prevent forward slippage. So at +1 frameshift sites, the program +1 frameshift can occur at its normal programmed rate. However, upon diphthamide deletion, -1 frameshifting is more likely to occur. So essentially what I think is maybe the +1 frameshift and the -1 frameshift are almost kind of like at a tug of war with each other. And this can cause more likely that there will be a net overall 0 frameshift upon diphthamide deletion. And so overall, this kind of suggests that diphthamide may actually increase +1 frameshifting as opposed to cells that don't have diphthamide. And so to further explore this idea, I plan on conducting some dual luciferase assays where I will insert various different program plus one frame shift sites and measure the amount of frame shifting in diphthamide and diphthamide knocked out cells. And this will help to further define exactly how diphthamide influences different plus one frame shift site sequences. I also further want to explore how diphthamide affects the cellular proteome as a whole. And to do this, I will conduct thermal proteome profiling, where I will compare, once again, yeast that are either wild type or DPH knockout. I will then heat these cells at a variety of different temperatures and extract the soluble protein. I will then label each different temperature with a different TMT label, like I mentioned before, and run these on the mass spec. From this, I can quantify thermal stability curves for each protein, and proteins that have a change in their thermal stability between wild type and knockout will indicate proteins that had a change in their protein-protein interaction status upon deletion. And so the TMT abundance data that I presented earlier to you gives you an idea of how diphthamide affected the translation levels of given proteins. This will show how those changes in translation levels then go on to affect the entire protein-protein interaction networks within the cell. And lastly, you may have noticed that my volcano plot for the DPH3 knockout cells had increased levels of significant proteins. And this is consistent with prior data that has shown that DPH3 may have alternative functions in the cell besides just making diphthamide. And one of those is that DPH3 interacts with the elongator complex, which has functions in making tRNA modifications. And so to further explore alternative functions of DPH3, I will use enzymatic proximity biotinylation with turbo ID to identify protein-protein interactors of DPH3. And with that, I would like to thank my thesis advisor, Amy Weeks, for all of her help and support with this project, and to the rest of the Weeks Lab, and to the biotechnology training program at UW-Madison for their funding and support. And with that, I'd like to thank you all for your attention, and I'd be happy to take any questions.
Great. Thank you, Lauren. So remember, if you have any questions, make sure to type them in the Q&A. Maybe while people do that, I can start off. So Lauren, in the mass spec, did you detect any new peptides? Right, so rather than proteins going up or down with known amino acid sequences, you can imagine a frame shift giving you a protein that has a new amino acid sequence, given its canonical one. Did you detect any of these new peptides?
I started to look through that, but I guess this initial search I just used. So when you do a mass spec data search, you have to input a FASTA file. And so for this, I just had used the basic yeast FASTA file for the whole proteome. And so, yeah, that is definitely another area that we would be interested in looking into. We would just have to figure out the best way to search for basically generate a FASTA file that would include various different frame shift sites that are non-canonical and not really reported in literature yet.
Since you have the wild type and knockout, can you subtract one set of results from the other to see which of the peptides are different, or is that too simplistic?
So for this, we can actually get abundance values, so it's not simply presence or absence. We can actually get like more exact quantification values to get more exact like ratios of a given peptide. Yeah.
Yeah, I guess I'm thinking to identify peptides that aren't even there in the wild type sample
I see.
because there was no frame shifting.
Yeah, because these are all, yeah, pulled together with the TMT approach, I think that would still, those would still show up because the TMT approach also helps minimize issues with missing values because basically all of the things will be there together. So even if it wasn't present in one sample, it should still show up in the data. Yeah.
Great. So hopefully see if anyone in the audience has any other questions. So feel free to type those in the Q&A box. Give people a minute. All right. So if there are no further questions, let's thank Lauren again. And we'll move on to our final speaker. So our final speaker is Saeed Roschdi, who is a graduate student also in the Biochemistry IPIB graduate program here at UW-Madison, and he is in Sam Butcher's laboratory. So take it away, Saeed.
Hello, everyone. My name is Saeed Roschdi. I'm a graduate student in Sam Butcher's lab, and today I'll be presenting my work on the conformational variability of poly-UGRNA. This work started back in 2019 with the discovery made by Mark Wicken's lab here at UW-Madison that showed that the C. elegans enzyme, RDE3, is a poly-UG polymerase, which adds pugtails, which can be either poly-UG or GU, to the 3' ends of mRNAs. This protein was of particular interest because previous studies had shown that it was essential for silencing activity in C. elegans. It was then shown that pugtails can permanently induce gene silencing in C. elegans. How this is done is that they've made a mutant of this enzyme RDA3, which prevents the normal initiation of silencing, which is occurring using double-stranded RNA. And then they can inject C. elegans embryos with GFP mRNA fragments with different 3' tails. And what they saw is that the only modification that inhibited silencing was the addition of this poly-UG tail, which you can see by the loss of fluorescence activity, which is not seen for any of the other dinucleotide repeats. They were then able to show that the silencing activity is transgenerational, seen by the maintenance silencing up to 3 to 4 generations after the initial injection occurred, and they were able to develop a model for how this is occurring. So the initial steps is the initial targeting of the mRNA of interest, which will then recruit the first round of Argonaute proteins, which are used to cleave silencing targets. They then will cleave the target mRNA of interest, and then RDA3 can then add pugtails to the 3' end of these RNAs. This then allows the recruitment of an RNA-dependent RNA polymerase to then make small RNAs that can then target the next copy of this mRNA fragment and repeat the cycle. And this cycle of cleavage and amplification will allow the transference of this silencing activity to future generations. We then became interested in determining whether these pug repeats are structured and if this structure has a role in silencing. And so to do that, we use circular dichorism to characterize the secondary structure features of different lengths of pug RNAs. And what we saw is that with these shorter lengths of 10 or 11 repeats, we see very little CD signal, suggesting that they are entirely or mostly unfolded. But when we add one more G and get to 11.5 repeats, we then see robust CD signal with a negative 240 peak characteristic of RNA quadruplex formation. We were then able to show that if you increase it to the 4-12 length, or if you flip the sequence, this structure is maintained, and that the structure can be informed internally. As a control for quadruplex formation, we then wanted to see if it can be formed with only lithium, since previous studies have shown that quadruplexes can't form independent of just lithium. And so if we look at this purple line, we see that there's little to no CD signal, suggesting that this is unfolded. In collaboration with the people who did the previous sciencing work, we were able to show that the length dependence of the silencing in vivo matches exactly the same length of P's as we see for our CD signal in vitro. This gave us some idea that potentially this structure might be playing a role in silencing. This was then confirmed using our understanding that this structure is a quadruplex by using an RNA modification where we switch out standard guanosine to 7-deazoG. And what that does is that this converts this nitrogen to a carbon, which then inhibits quadruplex formation. And we were able to confirm this both by circular dichroism as well as native gel electrophoresis. Using this modification, our collaborators were then able to go back and do the in vivo silencing assays with GFP well. When you do no injection, they see no loss in GFP expression. And when you do a normal pug, GFP levels are significantly silenced. But when you do the 7-deazoG modification, which inhibits quadruplex formation, but wouldn't affect standard Watson-Crick base pairing, you see no silencing activity, showing that this structure is essential for the silencing activity. We are then able to solve a high-resolution crystal structure of the structure we call the PUG4, which is a left-handed intermolecular quadruplex with a mix of 3 Gs and 1 U quartet. We were then able to show that these PUG4s can tolerate AA insertions done first in vitro using circular dichroism. We were able to show that an AA insertion into this GU12 RNA did not change any of the major peaks of the PUG4. And interesting enough, when you injected these modified RNAs back into the C. elegans silencing systems, you see that they are still able to induce silencing, meaning it's not just sequence composition, it really is further proof that it's the specific structure that's promoting silencing. We were then able to show that this PUG4 can form and transform 2 different strands of RNA using this short PUG repeat GU6, which is half the sequence length of the normal PUG sequence, which is GU12, and has all the same characteristic CD peaks, showing that we're able to get an intermolecular quadruplex PUG4 using 2 strands of GU6 versus 1 for the previous structure we solved. This then led us back to the silencing data. So we were able to show that this sequence is essential and enough to induce silencing. But if you look at the length dependence of silencing, you see it significantly increases as you increase the length up to around 18.5 repeats. So this led us to ask the question, is there some other structure that's also forming alongside the PUG4 that promotes this activity in these longer PUG sequences? So we then did circular dichroism for multiple different shorter repeats, GU3, GU4, and GU5, and saw that in a concentration-dependent manner, we were able to get the formation of another quadruplex structure, seen by the fact that these 260 and negative 280 peaks have been previously shown to be present in intermolecular quadruplex, and these structures do not form in lithium. And what's particularly interesting about these structures is that if you look at the original CD signal for the PUG4, you'll see a negative 240 and positive 260 and 280 peaks, which are exactly inverted in this GU3 structure, where you see a positive peak at around 240, 250, and then negative 270 to 290 peaks. This inversion of the CD structure suggested to us that this might be an opposite-handed structure. We are then able to show that at higher concentration, Gu6 is also able to form this new right-handed structure. We then looked at the concentration dependence of the right-handed G4s and see that as you increase the length of the RNA from 3 to 4 and 5 repeats, you see an increase in the binding self-affinity of the RNA to themselves, showing increased stability as you increase the length of these short pug repeats. We were then able to solve a high-resolution crystal structure of a GU RNA. This structure is a right-handed asymmetric homodimer of tetramers. So how does that work? You start with the basic unit cell, which is 2 strands of GU3 that contain head-to-head stacking at the 5' ends. This is then expanded by 4-fold symmetry to get this 8-stranded structure, which is made of 2 4-stranded quadruplexes. What's particularly interesting is because of this high resolution, we're actually able to capture some snapshots of RNA dynamics in this structure, seen by the 2 different phosphates that are present both between this GNU and between this GNU. You can see that there are 2 clear phosphate positions defined in this structure. If we then look through one of the monomers of GU3 at a base-by-base level, we see that the first layer of this quadruplex is a G1 quartet with U2s bulged out. We then see an inversion here that transitions us to a G3 quartet with a water bound in the center, a U4 quartet with a potassium, a G5 quartet with another potassium, and then we see the second inversion at the structure at U6, which then has a U quartet as well as sodium ions bound to the surface. What's also interesting, we see another example of conformational heterogeneity in that we actually, each of these monomers have 2 different G3 quartet layers. And what I mean by that is, in this top structure, we have G3A, which is not capable of, it's not bound to a water and the sugars in the C2' end of conformation, while in the second monomer, we have a water bound and the sugar pucker is the C3' ender. The reason this conformation changes the water binding so much is it actually increases the distance between these 2 oxygens as well as the center of the quartet, which in the one that doesn't bind is 3.9 angstroms between these and roughly 3 angstroms to the center, while that goes down to 3.4 angstroms for the second conformation and 2.8 angstroms for this, which is able to bind water. This allows us to show that these pug RNAs can form unique and different RNA quadruplexes depending on their length. So what we see is that for shorter repeats, in order to get enough quartets to form a stable structure, you need to use 4 strands, but this increase and have a longer overall structure. But the fact that it's multi-stranded allows you to have more bases in a quartet with only a U2 bulged out here, while for the original pug form, because it's more compact and is single-stranded, you have to have bulges and loops to connect the different strands together. This also affects the overall handiness of the structure, where because there's the steric constraint on the GU3 structure, it can form a more standard right-handed orientation, while for the original pug form, it forms the more unique left-handed conformation. This new structure is particularly interesting because recent studies have shown that RNA quadruplexes can form granules in vivo, both for long quadruplex forming sequences as long as short sequences that can form condensates in vivo that have a more solid-like property seen from these fluorescent recovery after polarization experiments where they don't fully recover to the initial fluorescence activity, both for the long and the short pug sequence. This then relates back to our pug repeats because silencing in vivo happens in mutator foci, which are perinuclear granules in C. elegans that contain Mu2, which is another name for RD3, an RNA-dependent RNA polymerase essential for PUG-induced silencing. So it'll be interesting to see what potential role these PUG repeats or the quadruplex structures have on granule formation in vivo. I'd then like to discuss some of the conclusions we were able to make. This is the first example of a left-handed quadruplex. This also shows that quadruplexes don't need to be consecutive Gs, as had been previously predicted in the literature. We see that these short pugs can form multi-stranded quadruplexes, and from these simple, low complexity UG repeat sequences, we can go to a variety of complex structures, and that the intramolecular pug forward marks RNAs as templates for gene silencing. I would then like to thank all the members of the Butcher Lab, as well as the collaborators for the help on this project. Thank you, and I'd like to be happy to take any questions.
Wonderful, Saeed. That's an amazing 1 angstrom structure. So we have one question so far in the chat. So Junji Joe or Guo says, beautiful work. Beyond the AA insertion, how many intervening sequences can a pug fold tolerate? Do the previously developed G4 ligands bind to either of the two forms of the pug quadruplex?
So we've not yet looked at any longer insertions. We have done a lot of individual mutations, which show that a lot of the U's can be switched for essentially any other of the nucleotides, but I've only done individual mutations. I haven't done multiple mutations of different positions. I've just done one mutation of all the positions. And we haven't done any longer insertions. And in terms of quadruplex ligands, we do know that NMM and methylmethylpropyl 9 binds to this structure. It is what was used to solve the original pug-forward crystal structure.
Great. Thank you, Saeed. So let's see if there are any other questions. I have a question, and I apologize if you showed this, but are the two different folds, what is their relative stabilities? Like, have you done melting curves? Is one more stable than the other?
So we haven't done melting for the multi-stranded structures because they're so, I guess they're highly concentration dependent. And so we assume they should be much less stable because you need to have very high concentrations in the hundreds of micromolar range.
Mm-hmm.
But we haven't done any thermal studies with them yet, just because the concentration requirements, at least for CD, make it hard to have the correct, as the detection limit, they're too concentrated. And so to do CD, you have to use cuvettes that you can't do melts in, really, because they're demountable. So it'll leak out. So we haven't done melts yet, but it should be less stable.
Great. So we'll give people another minute or so if they have any lingering questions. I'm not seeing any pop up so far. So I want to thank all of our speakers, Karli, Lauren, and Saeed, for sharing their work with everyone here. And with that, I would also like to thank you for joining us, and thank you to the organizers of the RNA Collaborative Series and the RNA Society for helping publicize and sponsor all of this. So thank you very much.